Thank you for the introduction. As Dan mentioned, I'm a postdoc in the fungal group, and more recently we've been sort of growing our interest in algal genomics. And so today I'm going to be talking about one of our projects where we're doing a multi-omics approach for studying a new green alga for the purposes of biofuel production. So at JGI, our efforts for interest in algal genomics is growing. And one reason why we want to study algal genomes is because algae are extremely diverse and yet largely unexplored. So algae have a very interesting and complex evolutionary history. And then from a practical point of view, there's also a lot of biotech applications. So all algae perform photosynthesis. And from a biofuel perspective, we're interested in how biofuels or how algae can produce lipids for biofuels. In addition to lipids, algae can also produce high-value commercial products that can be used in cosmetics or nutrition supplements. And commercial production of biofuels, though, has been somewhat limited to only a few number of algal species. So we're interested in exploring the potential of new algal species for biofuels. So what makes a good strain for biofuel production? Often we consider the lipids, and this was mentioned in Mary's talk in the first talk in this session, the triacyl glycerols, which are the precursors for biodiesel. And some algae can produce a high content of these tags under stress conditions, but there's this trade-off between growth and lipid production. And so for producing a high levels of biofuels, we have to consider how to maximize both growth rate and lipid accumulation. Additionally, for commercial production of biofuels, we want to grow these in these large outdoor ponds. But if you're growing these outdoors, you have to consider the environmental factors that could impact growth. So things like temperature could influence how much an algae grow or how much lipid is produced. And so how can we at JGI help to produce better strains for algal biofuels? So what we're doing is we're collecting multi-omics data of algae that perform really well in these screens, high growth and high robustness, and help them to characterize these algae to further improve them by strain engineering. So this project is a collaboration between us and Los Alamos National Lab. It's a Bioenergy Technologies Office funded project. And our goal here is to develop a functional genomics pipeline for algae for accelerating commercial biofuel production. So our first target is this strain of Senodesmus. It's a strain coming from the National Renewable Energy Lab, and it was shown to perform really well in these growth screens, in particular in the summer cultivation studies. And so our objectives for this are to produce the genome of Senodesmus. And in addition to the genome, we're also collecting the metabolomics data and transcriptomics data. And by integrating this data together, we can identify gene targets for strain improvement. So this is an overview of the steps in our functional genomics pipeline. It includes our standard genome sequencing pipeline for sequencing, assembly, annotation, and analysis. And then in addition to this, we do perturbation studies, and we profile the algae using transcriptomics and metabolomics under this perturbed state. But first, I'm going to talk about the newly sequenced Senodesmus NREL 46B D3 genome. So Senodesmus belongs to the class Chlorophyceae. That means it's related to Chlamydomonas. It's probably the more well-known algae. And so Senodesmus is also more closely related to some of these other oleaginous algae. So these oleaginous algae also produce a high level of lipid content, the Monorophidium neglectum and the Chromachlorus phaingiensis. The genome assembly of this Senodesmus strain was found to be 150 megabases, and about 17,000 gene models were predicted. So this puts it on par with its relatives. And now that we have the genome annotation, we can begin to identify homologs in the genome that participate in lipid metabolism. So here I'm showing the tag biosynthesis pathway, And then I have the number of homologs that were predicted in Senedespis as compared to Chlamydomonas for each step in this tag biosynthesis pathway. And you can see that there's a few additional copies found in some of these later steps of the pathway in the Senedespis as compared to the Chlamydomonas. And we can do this on a larger scale by building a metabolic reconstruction. So on the genome scale, we use automated tools to map genes to metabolic pathways to help us interpret this data. So next I'll move on to the perturbation studies where we profiled the algae using transcriptomics and metabolomics. And so these temperature perturbations were used to mimic the temperature changes in Mesa, Arizona. And these are the locations of the algal test beds. So at Los Alamos National Lab, Tiza, working with in Sean Starkenberg's lab, grew up these algae under cold shock and heat shock. So these are 5 degrees colder than the coldest outdoor temperatures observed in Mesa, Arizona, and 5 degrees hotter than the hottest observed outdoor temperatures. And then she also grew up 2 controls. And this was sampled over the course of 1 day, taking 6 time points. And then the light was controlled on a diurnal cycle. And then looking at the growth curves of the cold shock samples and the heat shock samples, over the period of sampling, we see that there's a greater change in the cold shock samples. So there was a slowed growth, whereas the strains that were grown in the heat shock samples tended to be more similar to the control. And we see these differences reflected in the transcriptomics data as well. So when we did a principal component analysis on gene expression, we see that the cold shock samples tend to be distinct compared to the control samples, whereas the heat samples tended to overlap with the cold shock sample, or with the control samples rather. And then also using the transcriptomics data, we built a gene co-expression network. So after filtering out low expressed genes and low correlation genes, we came up with 5,500 genes and about 1 million edges. Each of these edges represent 2 genes that are correlated in expression. And then you can see in this overall network structure that there are 2 distinct groups. So this top group up here was increased in expression in the cold samples. And then these genes down here were decreased in expression in the cold samples. And the differences in the heat shock samples didn't seem to drive any sort of network structure. Furthermore, we clustered the genes by expression, and these clusters are indicated by the colors in this network. And we performed functional enrichment analysis. So some of the Go terms that are relevant to growth or lipid production I've included up here in the colors associated with the corresponding gene cluster. And I'm going to focus in on 2 of these clusters. The 2, one of these is in green and one's in red, and they were both enriched in fatty acid biosynthesis genes. So this is related to tag biosynthesis since fatty acids go into the triacylglycerols. So here I'm showing a heat map of the differential expression of both of the clusters that were enriched for fatty acid biosynthesis genes. The red is indicating an increase in expression relative to the control, and blue is indicating a decrease in expression relative to the control. And if it's kind of hard to see, the overall gist is, like I showed in the network structure, these are increased in expression in the cold samples, and we don't see much of a pattern in the heat shock samples. I'm also showing, in addition to the lipid metabolism genes that were found in this cluster, there are also genes related to carbon and nitrogen metabolism. And these could be important regulators for carbon partitioning and related to the control of biomass production. Additionally, I'm going to show you the gene profiles of fatty acid desaturases found in these gene clusters. And we've also identified transcription factors that are co-expressed in these gene clusters. So these transcription factors could be potential regulators of genes within this cluster. So we've proposed that these transcription factors are potential candidates for strain engineering. Furthermore, we looked at the lipid levels and how these change in response to temperature. In addition to the triacylglycerols, I'm also showing you the monogalactosyl diacylglycerols and the digalactosyl diacylglycerols, which we also measured the levels of. So these are membrane lipids found in the thylakoids in chloroplasts. And you can see the changes in abundances in the triacylglycerols, the tags, is broadly increased in both the cold shock and the heat shock samples. Whereas we do see some temperature-specific changes. So for example, we see that lipids with a lower degree of saturation or a higher number of double bonds are increased in abundance in the cold shock samples. And then vice versa, we see that lipids with a fewer number of double bonds are decreased in levels in the cold shock samples. So it's thought that these changes in lipid levels and lipid composition is related to a cold adaptation strategy. And this has been observed in other plants and algae. If we go back to our gene expression data, I had pointed out these fatty acid desaturases, which were shown to be increased in expression in the cold shock samples. The fatty acid desaturases are responsible for adding double bonds in those acyl chains of the fatty acids. So we think that this relates back to what we see in the changes in the lipid composition. And this has also been previously observed in other plants, algae, and cyanobacteria. And then we also are proposing these fatty acid desaturases as potential candidates for strain engineering, since we know that lipid composition can have an impact on biofuel properties. So in conclusion, we've developed a new functional genomics pipeline for profiling algal strains. Currently, we're working on our second strain, Monoraphidium minutum. And this was identified in a Discover project, so they screened in winter cultivation studies and looked for high growth. And so that will be interesting to compare to Senodesmus, which was growing well in the summer conditions. And we're really trying to speed up the throughput of this pipeline. So we're trying to accomplish this on a 1-year timeline. So this would be twice as fast as our first strain. And then in the case of Senodesmus, we've observed some large-scale changes in the transcriptomics and the metabolomics data. And then we've identified several gene targets that we've proposed for strain engineering. Currently, these targets are being tested at NREL, and we're really excited to see what results come out of those. And then lastly, I just want to again thank my collaborators and all of those on the JGI team who've also contributed to this. Los Alamos National Lab has really partnered with us in this study, and REL has provided the strains and are doing the validation. And then I want to thank the organizers for giving me the opportunity to speak today, and then thank you for listening.
Thank you, Sara. Questions from the algal aficionados in the audience? So what are the prospects for strain engineering? How have these kinds of changes been attempted, and what level of confidence do you have that it's going to be translatable?
Yeah. Sure, these have been at least attempted, and of course there's been a huge interest in algal biofuels, and the thing is about how to bring down cost is the major factor. And oftentimes when you manipulate something involved in the lipid biosynthesis pathway, it has an impact on growth, like I mentioned, in the stress conditions. But there has been more recent interest and some progress being made to improve them. So I think, yeah, there's definitely a potential.
Okay. Thank you.
Mm-hmm.
Thanks, Sara.